Good morning, IB Bio students. The purpose of this video is for me to give you an overview of class today, as well as to get you set up for the lab activity that you're going to be doing. As you can see from the slide, we have three main objectives for today, and they are all linked to continuing our study of the movement of materials within plants. We're going to continue to work on our understanding of how water moves within the xylem of plants, and connected to that will be the lab activity where you're going to learn a technique for calculating the distribution of stomata on the underside of leaves, and then you'll use that technique to design a lab activity of your own. And then finally, you will begin your study of how phloem in plants transports nutrients like sugar and other complex organic molecules. You can see from the agenda here that a lot of the class period is devoted to looking at the transport of water again, and a little bit of time looking at the transport of nutrients. We won't actually spend time going over the quiz together since I'm not there with you, but I'd encourage you a little bit later in the period to get onto Canvas and check out the feedback that I left for you there regarding your quiz results. We are going to look at the transport of water and do a little bit of review together. Then you'll spend a significant part of today's period doing the Stomata Lab. Then you'll have some time at the end to begin your study of the transport of nutrients and continue working on the transport assignment that we started in our previous class period. We're going to begin our study of the transport of water by watching a very short video clip. This video is not the most high-tech in terms of graphics and so forth, but it's a very nice short overview of what we've talked about so far. So that's why we're going to spend some time looking at that together.
Plants transport water and nutrients from their roots to their leaves through a vascular tissue called xylem. These are dead, empty cells which connect end to end, forming long continuous tubes. Because water molecules carry a slight electric charge, they're attracted to each other. This natural attraction, known as cohesion, links the molecules in long continuous chains. At the leaves, water escapes the xylem and evaporates through microscopic pores on the surface called stomata. As each molecule breaks loose, it pulls up another molecule to take its place. This continuous pulling tension extends all the way down the xylem tube to the roots. Here, the rising chain of molecules keeps pressure low, allowing water and nutrients to enter through osmosis. Tissue called xylem.
So as you heard in the video, and as we discussed in our previous class period, water moves through the plant from the roots, through the stem, and eventually exits the leaves through openings called stomata. Those stomata are responsible for allowing water vapor to escape. They're also responsible for gas exchange. So the plant will have stomata open so it can take in carbon dioxide and release oxygen to generate or to fuel the process of photosynthesis. So we're going to look a little bit more detail at these stomata here in just a moment. But before we do that, I'd like us to spend just a little bit of time thinking a little bit about what all we remember about this process of transpiration and the role of stomata in transpiration. So take a moment to pause the video and with your seat partner, discuss the role of stomata in the transport of water into and within a plant. And this could be a test question that would be worth 6 marks. Now that you've had a little bit of time to discuss with your seat partner, here's what the mark scheme looks like. Take a moment to look at this and to determine how many marks you and your partner could have received if this were actually a test question. Okay, so returning to this idea of stomata and the lab activity that we're going to be doing today, the major purpose is that you're actually going to learn a new technique for making an impression of the stomata on the underside of a leaf, and then for counting those stomata. You will then use this new information to design a lab activity where you will either look at the effect of factors that affect the distribution of stomata on plants or factors that affect stomatal opening and closing. So that will be the second part of your lab. If you look at the pictures here on the screen, the one on the left is a cross-section of a leaf that you looked at under the microscope, and the openings at the bottom would be the stomatal openings. The other picture on the right is what your slide today is actually going to look like. And so those circular structures that you see there are the cells that surround the oval openings that are the stomata. But just a little bit more background information about stomata before we take a look at the lab itself. We know that stomata regulate the passage of water. That also regulates the passage or the exchange of oxygen and carbon dioxide. On either side of the opening are two cells that are called guard cells. And those guard cells literally do what the name implies. They guard the opening. They regulate whether the stomata is open and water and oxygen and carbon dioxide can pass through, or whether the stomata need to be closed so that those processes stop. This is a really great example of the marriage between structure and function. Guard cells are shaped in an oval pattern, and the inside, the side towards the stomata, has a very thickened cell wall. So what happens is when there's a lot of water present in the plant, water will diffuse into the guard cells. This causes them to get very turgid, and they actually bend, and they bend unevenly because of the thickening of the cell wall. And so that causes the opening of the guard cell. When there's not water, a lot of water present in the plant, the water diffuses out of the guard cells. That causes them to shrink, and you can see that the guard cells then close again. This is kind of an ingenious process because we want the guard cells to be closed when there's not a lot of water present in the cell. When there is a lot of water present, then we can have the guard cells open the stomata so that photosynthesis can occur. There are a variety of different things that really impact the opening and closing of the stomata in addition to water. Light from the blue portion of the visible spectrum actually causes potassium ions to diffuse into the guard cells. It's this movement of potassium ions that actually draws the water in and opens the stomata. Then at nighttime, when photosynthesis doesn't need to occur, blue light isn't present, so the potassium will actually diffuse out of the guard cells, and that will also result in the closing of these cells. I'm going to show you another very short video clip here that kind of emphasizes this relationship and gives you a little bit more detail on the opening and closing of the stomata. The important thing is to understand that it's really all driven by light because it's all driven by the process of photosynthesis.
About 95% of the water absorbed is lost through the stomata present in the leaves. This process is known as transpiration. The number of stomata is more on the lower surface of leaves. If transpiration happens throughout the day, then there will be a great loss of water. This is controlled by the stomatal opening and closing mechanism. During the daytime, water enters into the guard cells along with the potassium ions. This brings about a change in the shape of the guard cells, and the stoma opens. During the night, the potassium ions and water molecules leave the guard cells. Thus, the turgidity decreases and the stoma closes. Stomata also act as main sites of gaseous exchange in plants. Leaves make use of water and the carbon dioxide for photosynthesis. Glucose formed during photosynthesis is transported to the other parts of the body through phloem cells.
So hopefully that video and some of this discussion has given you a better understanding of how the stomata open and close and their role in photosynthesis and water transport within the plant. We're going to shift gears at this point to looking at the actual lab that you're going to do. I'm going to go over just a couple things here with you at the very beginning. I'm going to show you a few things and then turn you loose to actually get started on it. You will be given a set of instructions, and it's really important that you follow these instructions very, very carefully. I'm going to demonstrate a couple of the key points, but I'm going to rely on you to really read and follow the directions carefully. Your end goal for the first part of this is that you are going to actually calculate the number of stomata per square millimeter of leaf tissue. So you're going to go through several calculations. These are laid out for you in the instruction sheet, and there's a place for you in your Google Doc Lab report for you to be able to show your work and show your processing until you get to the final answer. Today, when you're finished with your slide, you're actually going to turn it in to me, which is something that you haven't done before. And then you're going to choose one of the 2 extension activities with your partner. You're going to design your second idea for a lab that you could do that incorporates this technique that you learn in the first part of the lab. By the end of the period today, you should be done with this lab. And so make sure that you've shared your Google Doc with me. And then there'll be a few things that you can work on in the time you have remaining that I will talk about here in just a moment. You are all going to be using portions of a leaf from this plant that you can see in the back of the classroom. And at this point, I'm going to actually demonstrate a few of the things that you're going to do with this lab. So make sure that you have your directions in front of you and make sure you follow these instructions really, really carefully. One of the important things is that you only need a really small portion of a leaf. So all you need is a really small piece. This is a couple of centimeters by a couple centimeters. The other thing is that we know there are many more stomata on the underside. So make sure you know which side is the bottom, and it's generally the side that is a little bit less colorful, a little bit paler in color. One of the key things that you need to do with this is you are actually going to take fingernail polish, clear fingernail polish, and you are going to paint most of the bottom of your piece of leaf tissue. So I know you probably can't see that very well, but I just painted a very small, thin layer of clear fingernail polish on the bottom of this. Then here's a key portion of the lab instructions. You have to let this dry completely. The instructions say at least 6 to 7 minutes. So I would set that aside. At that point in time, you can access the Google Doc that I've shared with you. You can get that set up. You can read through the background information if you haven't done so already, but definitely make sure that you let that dry completely. This is the step that will cause you to start all over again if you don't do it properly. So I know 6 to 7 minutes haven't passed, but I actually did one earlier that was completely dry. Your next step is that you need to actually lift this impression, this layer of dried fingernail polish with very clear tape. And it's really imperative that you do this without getting fingerprints on the tape to the best of your ability. So you want to peel this off, and you want to peel a little bit longer than what you actually need so that the portion you're touching has the fingerprints on it, but the portion you're not touching can be laid right on your fingernail polish. And so you're going to press this down as carefully as you can. You might want to use some lens paper to do the pressing so that you're not putting fingerprints directly onto your piece of tape. And then you're going to very carefully peel the tape off. And as you can see, the fingernail polish came off with it. You can also possibly see my fingerprints there that you don't want to look at under the microscope. Then you're going to get a brand new slide. And the slides you want to be very careful that you just handle the edge of. So again, that you don't put fingerprints on this because fingerprints will make the stomata a little bit more challenging to count. So you're going to very carefully lay this down and do your best to not get wrinkles on it, to get it as flat as possible. You can take the end of a pen or a pencil eraser and kind of smooth it down. You can use lens paper to push it down. So again, you don't put fingerprints on it. Then the extra here that has my fingerprints on it, I can just cut off. And I'm ready to look at this under the microscope. And then you'll have to follow the directions in the lab for counting these and for doing the calculations. You are only making one of these slides, but you will look at 6 different spots on here. If you're in a group of 2, you each do 3 of the counts. If you're in a group of 3, you each do 2 of the counts, but you'll end up with a total of 6 counts, but you do not make 6 slides. You just move the slide to 6 different places and try to do that in a systematic fashion so that you don't count the same spots over and over. Be very careful to read the directions and know what power you are looking at these under. That makes a big difference. You will also actually have to measure with a ruler the diameter of low power on the microscope. So that involves putting a ruler directly on the stage and then looking through the objective and counting the number of millimeters there. That's also an area that can become a little bit tricky to do. And make sure that you estimate if you don't have an exact number of millimeters so you have as much accuracy as possible. When you're finished with your slide, then you'll get one of these labels, put both your names on it and your period number, and then you'll turn it into this slide box that's up here so that I can see your slide that you've created. So those are the instructions for the lab procedure. Hopefully you'll be really proficient at following the directions, and we'll come up with a nice slide and you'll have some good data to use. When you finish with all of this, including the extension, again, make sure you've shared the Google Doc with me so I can take a look at it. And then in any time that remains in the class period, you should go to Canvas and access the 9.1 and 9.2 plant transport video part 1. And you should watch that and listen to that. And in that video, you will learn about the structure and function of phloem. The first 5 minutes of the video are actually a review of the structure and function of xylem. So you're welcome to watch that if you want to review that. But if you want to jump right to the part on phloem, you can jump to the 5-minute mark. I would encourage you to have your notes that go along with the transport assignment in front of you and go to part 3 and just kind of follow along those notes about the function of phloem. And after that, then you should use the remaining class time that you have to work on your transport assignment. Hope you have a great class period.